anaphase 2. So let's go back. So I like this figure personally. So if we were looking at anaphase 1, sometimes you can confuse anaphase 1 with 2. You still have the sister chromatids attached. Homologous chromosomes separate, but the sister chromatids still attached. And here, this is where they separate in anaphase 2. So that's the answer. Before I move on, are there any questions about meiosis or what we covered so far, about any of the vocabulary words that we covered today? Yes, no, maybe? No, okay. So next thing I'm going to cover is called crossing over. Crossing over, two homologous chromosomes exchange genetic material during prophase 1. So what you want to note from this is crossing over happens in prophase 1, and crossing over is exchange of genetic material. You have two homologous chromosomes. You can see the genes on the chromosomes, and when they come close to each other, this is what happens. That's called crossing over. And now this chromosome has some information from this chromosome, and this chromosome has some information from this chromosome. So that's crossing over. That's what gives you diversity. Goes through meiosis 1, this is what you got. Meiosis 2, this is what you end up with. Parental and recombinant. When you see the recombinant, means it's recombined. So some material comes from here, it gets recombined with this. Same thing for this one. So that's called crossing over. There is something else that I need you guys to know. That's called independent assortment. Independent assortment means the chromosome pairs, they're going to align randomly in metaphase 1. You don't know which chromosome is going to align with which one. So they align randomly during metaphase 1. So let's look at the alternative. So here you have 3 chromosomes. We're looking at 3 blue ones, 3 red ones. And it goes through meiosis. This is what you get. One cell has 3 blue, other one has 3 red. That's your first alternative. Then for the second one, this is how they decided to align the chromosomes. Now, when it goes to meiosis, this is what you get. You get 2 blue, 1 red, 2 red, 1 blue. And these are the sperm cells. You don't know which sperm is going to go fertilize the egg. So maybe a sperm cell has this material, another one has this one, another one has this one, another one has this one. And you don't know which one's going to go fertilize the egg. Depending on the one that fertilizes the egg, that's the genetic material the offspring is going to receive. In the third situation, we have, again, 1 red here, 2 blue on the sides. And after it goes to meiosis, this is your possible combinations. 2 blue, 1 red, 2 red, 1 blue, and they can be different chromosomes, and same thing, another alternative. So there are so many possible ways for the genetic material to get into each cell, and it's done randomly, and that's called independent assortment. Independent assortment. There's crossing over, and then there's independent assortment. Now, if you were to calculate the possible outcomes for these 3 chromosomes, you're going to, because they're diploid, so you're going to say 2, and then you're going to do 2 to the 3rd power, and that's 8. You have 8 different possible combinations. So a germ cell with 3 pairs of chromosomes has 4 possible arrangements during metaphase 1 and yielding 8 possible gametes. So we can actually calculate this for our cells, human cells, and we know that there are 23 chromosomes. So what you're gonna do, you're gonna take this 2 and put it to the 23rd power to get the possible, so to get the different possibilities. So if you have a scientific calculator right now, I want you to do that for me. So tell me what are, what's the number for the different possible outcomes for our chromosomes. So 2 to the 23rd power. It's probably going to be in millions or billions. So once you do that, put it in the chat. $8 million. So True said 8 million. 1, 2, 3, 4, 5, 6. So there is basically 8 million different possible combinations, and then the egg is going to get one of those. If someone has a child and 2 years later they decide to have another child, it's 1 in 8 million possibility that the same genetic material combination is going to fertilize the egg, or the sperm that has the same combination as the previous child is going to fertilize the egg. And that's why children with your first child doesn't look alike as the second child. I'm not talking about twins here. I'm talking about if you have a child today and then 2 years later you have another child, they're not going to look alike. And that's because of these different combinations. Does that make sense? Give me thumbs up if that makes sense. So now we're going to talk about twins. We said, so this is monozygotic or identical twins I'm talking about. Mono means one, zygotic means the zygote. You have one zygote. When it goes through mitosis, it splits. And now you have two embryos. Sometimes this happens. So monozygotic twins or identical twins, you have the egg and you have the sperm. The egg gets fertilized. Now you have a zygote. The zygote splits. Now you have two embryos or two offspring. And they're going to look the same. But that doesn't mean they're the same person just because they have similar genetic material. They're still going to have different fingerprints. One child might have moles in places where the other child doesn't. And then the life experiences count as well. So they're not the same person. They just have similar genetic material, but the fingerprints, they still differ. There's this case I want to talk about a little bit. I don't know if you guys heard about it. It's called 1 in a million. A twin too. Let's see. Here, this one. So have you guys heard about this before? So they had twins and one looked like this, the other one looked like this. Have you guys heard about this? Might, no? Yes, Nancy said yes. So it might be difficult to understand, but if you, so I think they said the child had Caucasian DNA. So if you think about it, the African American people, they were brought in as slaves and then they were forced to mate with their white masters. And there is some, DNA that's left in the ancestry. So that's how this might be possible. So depending on the combination you receive, you might have a child that looks completely different than the other child. But this is not identical twins I'm talking about. I think it's the next slide here we have. So this is the one I'm talking about. I'm talking about fraternal twins. They might look completely different. Sometimes you might see cases like this. So I think this, I think this is a really good example of genetics where, one child looks completely different than the other child. That's because this child received maybe a sperm cell that had some Caucasian DNA combination in it, or maybe from the mother, it came from the egg cell. So this we already talked about, monozygotic or identical twins. Zygote splits in two in early development, and then you have identical twins. But then we also have dizygotic. Di means two, zygote means zygotic means zygote. You have two zygotes. So in this case, they're gonna have variation because a different sperm is going to fertilize this egg, and different sperm is going to fertilize this egg. This sperm has a different combination of genes compared to this sperm. So because they went through random assortment, the twins, they're going to look different. They might look very different. So this is, one zygote makes one embryo, the other zygote makes the second embryo. If you guys know anything about female anatomy, there are two ovaries. and then the egg releases in the fallopian tube, and then once the sperm fertilizes it, it's gonna set in the endometrium and becomes the zygote and starts to go through cell division. Sometimes some females might release two eggs, maybe one from this ovary, maybe another one from this ovary when they ovulate, and then the both of the eggs are going to get fertilized and they're going to have twins. If somebody has infertility issues, they might give you a medication that's going to release multiple eggs each month, and it's going to increase the chance of having twins as well. Another thing I want to mention, sometimes what happens, maybe a female ovulates and from this ovary, and then she mates with a male, and the sperm comes in and fertilizes that egg. And maybe a little bit later, she releases another egg. She ovulates and she releases another egg, maybe from this ovary, and maybe she mates with another different person, and now this egg gets fertilized. So it is possible to have twins that are fathered by different people. So that is possible. So sometimes that happens. But today we're only talking about fraternal twins and the identical twins. And if that happens, if there are twins that are fathered by different people, they are going to look completely different.